Welcome to the Bioinformatics CRO podcast. I'm Grant Belgard, and I'm delighted to welcome Elliott Margulies, Director of Bioinformatics at BillionToOne, a molecular diagnostics company applying quantitative approaches to prenatal screening and liquid biopsy. He's spent his career at the intersection of genome technology, computation, and clinical translation, including earlier work at NHGRI and Illumina. In this episode, we'll talk about what he's working on now, how his career developed, and what advice he has for scientists and engineers who want to build bioinformatics that matters in the real world. Elliott, welcome to the show.
Thanks, Grant. It's really great to be here. Great to see you again, too.
The pleasure is mine. For listeners meeting you for the first time, what are you working on now and what problems are most alive for you day to day?
Well, I'm, like you said, at a molecular diagnostics company. And so day to day, my team focuses on the processing of data for prenatal testing. And we focus on everything from kind of the infrastructure and analysis of kind of primary sequence data, making sure the instrumentation is running correctly, our QCs are appropriate, all the way through to building, maintaining curation and interpretation systems and building out reporting platforms. We're kind of the glue across all the different parts of the organization that has to touch a sample from when it's acquired to when it's reported out, whether that be the lab, our genetic counselors, lab directors, even some software teams who are managing interfaces and portals that our physicians and patients interact with.
Would you explain the core bioinformatics challenge in your current work to a genomics audience that's new to this particular area?
Well, we very much focus on actionable, informative places in the genome. This is actually kind of a migration that I've made in my own career. From figuring out algorithms and methods to interpret and analyze whole genomes, bringing that to scale and thinking about how healthcare systems might adopt that, to now being at a company that focuses very much on very specific parts of the genome, where we are able to determine with high confidence whether risk variants or other deleterious variants are present. And we can do so both by looking at the DNA from mom, but we can also look at the cell-free DNA that represents the growing fetus inside of mom and determine the probability that the baby is carrying 0, 1, or 2 deleterious alleles.
You make technical work understandable across teams with very different vocabularies.
Oftentimes, we remind ourselves that everybody is in a silo to a certain extent with their own lingo, and we make it comfortable for people to pause and say, wait, I don't understand you, or what was that acronym you used? And we really have a fun culture of kind of acknowledging that everybody is an expert in their own field, but when those interfaces start to talk to each other, you got to slow down and not be afraid to say, I don't understand. It's actually a really strong part of our culture at this company right now, where that understanding of constant learning and explaining, or feeling comfortable saying, I don't understand, or feeling comfortable saying, I think I may have screwed that up. Let me fix this, or let me learn what happened with the process that we can improve upon. But it all starts with clear communication and feeling comfortable at those interfaces, and slowing the conversations down, up-leveling, using analogies.
What do you wish more people understood about the bioinformatics behind clinical testing?
We're incredibly rigorous, right? So when I think about the reproducibility and the confidence we have to have that all of our pipelines are working. We're constantly thinking about how can we cross and double and triple check that everything is working correctly, that we're giving the right answers to the right people all of the time. And so the running a pipeline is easy, but running a pipeline at scale with a level of confidence that every patient is getting the right answer as often as possible, and that we have ways of, because oftentimes it's the exceptions that we spend the most time on, right? So fortunately, in a prenatal testing situation, most samples are negative, and that's a good thing. Or I guess another way of thinking about it is that most of the time, sequencing runs and analyses work out just fine. But we spend the majority of our time on those small exceptions, whether it's a complicated case, a sequencing run that didn't go as well as we thought, some other issue that's kind of coming into the pipeline. We spend the majority of our time, right, thinking about, those small number of samples that are not within the norm.
When you encounter those edge cases, are they typically treated as one-offs, or do you typically make changes to the pipeline or process or something on the basis of those, or are they just so rare and weird that they typically don't trigger kind of?
No, I'm actually going to quote a team member of mine. His name is James Hart. We were recently talking about building new pipelines for future products, and we were talking, and I had said something to the effect of, we need to make sure we can handle the edge cases. And he looks at me and he says, as he has a great mindset when it comes to building these types of infrastructure, he says, "Elliott, there are no edge cases. There are only workflows that are run rarely." And it was a nice epiphany, I thought, where and I was like, oh, we've got the right person thinking about this. But it is true in the sense that for some of the legacy systems that we manage, when there are edge cases or one-offs, we see them with some level of repetition. And particularly as things scale, it's important for us to build systems that can more handle those types of situations in a more standard way.
I guess on that note, how do you decide whether a problem needs a better assay, a better model, better data, a better process?
Yeah, there are many types of rules and kind of prioritization, right? So you have, a finite set of really smart people on your team who are balancing both kind of production responsibilities as well as building new products, as well as automating and scaling kind of existing infrastructure. And we balance our time across these things. And oftentimes we look for, the proverbial low-hanging fruit, or take that 80-20 rule, where can I spend a small amount of my time and have that be a large impact? And oftentimes we think in that way when we prioritize work, particularly over the short term. And so we try and balance the short term. Where can we do a small amount of effort that's going to have a big gain? And another metaphor sometimes we talk about is those types of small efforts that have large gains start to get a flywheel going of more automation. And that more additional automation frees up even more time to then automate even further. And before you know it, you're starting to spend time thinking about the larger system and the longer term growth that is necessary.
What do you think about uncertainty when a computational result may affect a clinical report?
We're very much a first principles based company. And so a lot of the analysis that we do is rooted in statistics and probability and quantifying. We have the ability to essentially count molecules that are at a very low level. But we can do so in a very quantitative way. And so oftentimes we put probabilities around all of our calls. And we are constantly looking at outcomes data. So we have a fantastic outcomes team that, of course, it takes 9 months or so to get the outcomes, but over time these things build up, and we scrutinize our false positives, our false negatives. They're very, very small and they're rare, but this is a screening test that we are looking at, and those can lead to ways in which we can think about systematically continuing and to improve the assay.
makes a quality metric useful rather than ornamental?
If it drives at something that is measurable and changeable. And again, that's very much in our kind of ethos, is this concept of being guided by first principles. We oftentimes want to measure something directly versus indirectly. I think as bioinformaticians, it's easy to kind of pull together some sort of, convoluted, I'll call it convoluted correlation. But if you can't detangle, what are the values and metrics going into why you're seeing this correlation, it becomes abstract, and you don't know what you're missing or what you're not seeing. And so oftentimes we try and peel away the layers of the onion so that the analyses we're looking at are rooted in first principles. And we've seen successes in doing that. I can think of a recent example where we were doing an analysis, and we could draw a weird square around certain data points and kind of increase our sensitivity and with a minimal increase to no call rate. But at the end of the day, we actually found that doing a more principled z-score approach was much cleaner, and we understood what was going into that z-score in a much better way. And so that's constantly on our mind as we're doing these analyses.
Where do you see AI being genuinely useful in molecular diagnostics?
Oh, one question that I know is potentially talked about all the time, but even over the last 6 months, we are seeing bioinformaticians turn into full stack software developers, but with this scientific level of expertise where they deeply understand the workflow and the systems. And so they're in some ways hyper enabled to move things forward. We've gone from, building out prototypes that involve, tab delimited files and filtering them through Excel spreadsheets to now, on the first try, having a customized user interface, that is designed for the particular workflow that we're doing. And that's all empowered because we're historically not front-end developers, but we're now empowered to be able to be front-end developers with the assistance of AI. And so I've seen the aggressive but thoughtful adoption, both as a software development tool and also as a data analysis tool. So you can quickly advance a variety of different analyses with AI assistance. I think, again, we're hyper enabled because we're already on the command line. We're already accessing our databases and our infrastructure and our systems. So the access to the data is not a problem. The envisioning of what we want to do is not a problem. And so we can very well prompt and describe to AI systems what we're looking to do. And I've watched both on the software development side and on the data analysis side, the speed with which we can move is just incredible. You almost have to pause because a human being can't absorb it all as fast as we're able to generate it all, right? And that's actually, we recently had a hackathon as a team, and one of the comments was, yeah, I built this thing so fast, but I still need to pause and look under the hood and really fully understand, what was going on here. And that disconnect is to the actual line by line coding is what everybody is starting to get used to and try and work through.
What changes when bioinformatics moves from method development into high throughput clinical operations?
There's a little bit more of a rigor associated with change control, ensuring the quality of the output is what you expect it to be. In many ways, we strive for things to be as perfect as possible, but more importantly, knowing where it falls over and documenting that and having an appreciation for that is in some ways more important than getting the algorithm perfect. we never want to be blindsided by an error or an issue that we didn't realize our systems weren't performing as well as they should. And so we're constantly on the lookout for that from a clinical standpoint, particularly also as the scale comes about. But we have, fantastic team members who think through a problem, and anytime we're able to make an improvement, one of the exciting things is that we're sitting on a treasure trove of historical data that we're able to understand with outcomes information. If we make an improvement, how well does it perform? How does it compare to the way our previous version was with the system? And so we build automation in systems that are able to essentially do an A/B comparison, right, off of tens of thousands of samples, if not more, to be able to have that rigor whenever we push a new change into the system.
How do you know when a pipeline's ready to leave R&D and enter routine use?
The formal way is through an actual validation. We set up a multi-arm trial that kind of tests both kind of individual measurements as well, and then it grows from there to reproducibility to final kind of validation outcomes. At the end of the day, we're making a variant call or an assertion of risk, and we have to have a known truth set, and we have to show reproducibility, and then we also try and show, almost like forcibly show limits of detection. Whereas there's flexibility in the system where even if it's not perfect, it's a little bit noisier data, we're still getting the right answer. Like understanding that limit of detection is really important. But it all happens through, we follow very standard processes by which we verify and validate our algorithms.
Now to shift gears and talk about your career. What early experiences most shaped the scientist you became?
I can point back to a lunch I had as a grad student where Francis Collins was a professor of genetics at University of Michigan. He was now at the NIH, but he had still come back on occasion through his relationships with the University of Michigan at the medical school, giving lectures on occasion. And hearing him talk about his day moving the Human Genome Project forward is still a core memory of mine as a kind of a young graduate student. And that planted a seed in my head about how you can take a doctorate degree and not just be an academic professor writing research grants for the rest of your career. Kind of the desire to collaborate on a scale that was beyond what one person can do it started to really kind of be a seed that was sprouting in my brain. And then moving to the NIH for my postdoc with Eric Green, who I know you've recently had on this podcast as well, really opened my mind to that type of, at the time it was called big science, right? Where you could generate data at scale, you could collaborate at scale, and it was a ton of fun. I've constantly found myself moving in those directions where the learning from a diverse group of scientists feels much more comfortable to me than working on an individual assay or experiment or project on my own. I have those little pet projects here and there, but at the end of the day, it's watching the group of people accomplish something that was not possible as individuals. That's been a ton of fun for my career.
Can you tell us about some of the later major inflection points in your career?
Sure. So I sometimes think about my career, at least right now, as having kind of three major phases, I guess. So I started as a faculty member at the National Human Genome Research Institute, where I was doing more basic science research, but in this kind of largely collaborative way, trying to make an impact on the world with this transition from kind of population scale, low coverage sequencing to find variation in the human genome, to being able to sequence individual genomes. And how do you build the methods that can accurately call variants in individual genomes? I then, kind of the second phase of my career is when I moved to Illumina, working in this advanced medical research group with David Bentley and his team, where we really thought about both kind of balancing what is possible and from a clinical medical perspective at scale, and balancing that with kind of the drive of the next generation sequencing that Illumina at the time was driving forward. And it was a fun balance because we were trying to provide utility to the sequence data that was being generated and added value. And thinking about, I was able in that environment to think 5 to 10 years out. How can we build collaborations to show what's possible about bringing, high throughput whole genome sequencing to a medical system. At the time, we had recently started the 100,000 Genomes Project in England as a kind of a first initiative of bringing genomes into a clinical context. And then this third phase of my career, which I'm in right now, which to me, there's a bit of irony in the fact that for much of my career, I had spent focusing on whole genome methods for sequencing and interpreting genomes, to now sequencing super small parts of the genome that are incredibly actionable and meaningful, and doing that at scale, and being so close to patient care. This is something that is incredibly meaningful to me and my team. And I don't think it's often you get a bioinformatics group like we have here that when we find a high-risk result, it weighs on us. And we know that we're now supporting genetic counselors who are going to be giving this information to families at a very, at a very challenging time in their lives, but they're going to be better for it because they have this information. We really believe that having that information is empowering to families. But we are, as a bioinformatics group, so close to those decisions and supporting teams who are delivering this information. It really is remarkable. And in other parts of my career, even though I was building tools that were interpreting genomes or building methods that could identify variants in rare diseases, I was never this close to patient care. And it's very rewarding to be able to support an organization like this.
How has your view of sequencing changed as the field has matured?
Well, I think we've reached a point where sequencing is a commodity. I remember a time when it was very novel to be able to do massive short read sequencing. That is now a commodity, and it is just one part of a toolkit that enables clinical adoption. And something that I've learned a lot from this most recent part of my career is how much you have to think about holistically a sound business model that is reimbursable, and you're building a system that can operate at scale within certain cost measures, and where sequencing is just one part of that cost. But thinking about how to interact with insurance companies, with physicians, with patients, is all part of the process now. So it's become, it used to be, I guess, to answer your question, it used to be a major part of everything that I thought of as a bioinformatician, and now it's just one part of many that are equally, if not more important, because we've essentially succeeded in making DNA sequencing a commodity.
What have different work environments taught you about how science gets done?
It's all about the people and the relationships. I've been around smart people, I think, all throughout my career, whether I was in grad school as a postdoc, at Illumina, here at BillionToOne. And the times where we were successful and impactful were the times when we could get large groups of people working well together and feeling comfortable and open to making decisions under difficult situations, not being afraid to call out when there are issues or concerns or timeline risks or technical risks. And I can point to the exact opposite happening where sometimes great ideas didn't succeed because people were afraid to say, I don't think this is going to work, or I don't know if this other person agrees with this plan forward, right? And everybody can be well-intended, but if you don't have that open way of communicating and being able to talk about the more challenging things in a calm, open way, oftentimes you can't see these great wins and these great successes. And I found a company that truly values all of those parts of the scientific process and the development process. So I really feel supported, both my kind of senior colleagues as well as the team members that I'm mentoring, to foster this type of environment.
What's something you've learned the hard way?
I think finding an organization and a mission that is aligned with your own career goals and growth is important. I can think of a time in my career where I thought I was well aligned with where I was trying to move things forward, but it ended up not being the same as the organization that I ended up being in. And for a period of time, I really struggled, not because somebody was trying to set me up to fail, but because my interests and where I thought the organization wanted to go was different than where the people who were leading the organization wanted the organization to go. And it took me a while to realize that disconnect. And once I kind of realized that, and I was fortunate enough to be able to kind of move into a different organization that was well aligned with my goals, my career started moving forward again. And I think that's oftentimes, right? Nobody's ill intent, but when you get that mismatch of like where you want to go, what your passions are, and if that's not aligned with the organization that you're in, that can be a rough time. But to your point, I can also look back on that and say, I learned a lot during that time that I wasn't necessarily moving my career forward. In hindsight, it has helped me move my career forward because there was a lot I learned during those periods of time.
Related to that, what have mentors taught you that still shows up in your leadership style?
I can point to very specific things that I do that are relics, so to speak, of things that I learned because mentors have done them to me. And some of that is fostering that deep collaboration. So Eric can think about Eric Green working in his lab. He just opened doors for me to collaborate with people that I didn't know I could collaborate with. And I learned so much about that. And I enjoy when I can bring people of kind of from different parts of the company together and start to see their minds just go like, oh, wait, I'm thinking about this problem too. And they're thinking about it from a different angle. And you kind of see that collaboration take place. I can think about, as I move my team forward, and I constantly am trying to think about how are they happy? Are they growing? Are they doing what they want to do? When we get a chance to be together, recently we had our team's offsite, we all shared a success and a challenge, and giving everybody the space and the comfort to share a challenging moment that they might be going through with the entire team in a way that is very healthy and productive, and they don't feel like they're going to be shamed or disadvantaged because they shared something they're struggling with. To create that environment, it becomes very empowering. And that's something that David Bentley taught me. So I can think about, that type of interaction of leading a team that's very thoughtful and constantly trying to grow. And these are things that definitely come from great mentors that I've had in the past. On a lighter anecdotal note, music is really important to me. Francis Collins is somebody who always brings music to a lot of the science that he does and the way in which we kind of bring teams and communities together. I started my offsite by singing a song to everybody and having them sing with me. These are things that I often do that are fun to think back on how wonderful leaders and mentors have kind of touched my own life and career and how I'm trying to give it forward to the rest of the team that I now lead.
What do you look for when hiring bioinformaticians who will thrive in clinical diagnostics?
So we hire people at different stages of their career. And I've been very impressed with the tenacity and thoughtfulness of the right person who is kind of just coming out of an undergrad or a master's degree. The work that we do can be very impactful and has to be done with a lot of thought, because these are patients who may be getting a high risk result. And it takes a certain kind of individual who has a level of maturity to understand that what they're building has to be right as best as they can do it, where you have to own up to a mistake in a way that can be productive, where both you learn and we learn how to improve the system. So I think finding a smart individual who can code and run bioinformatics pipelines is kind of table stakes. It's really looking at an individual. What are they passionate about? How do they want to make an impact in the world? How do they want to grow their career? Are those passions aligned with being in a company where we are managing patient samples, being in a company where we're doing science that is leading to new products, where we're going to be balancing an intense period of time of scaling and building new products and trying to automate more, so that multitasking ability, but at the same time trying to ensure that people can focus and not get overwhelmed with everything that's going on. So these are kind of traits we try and look for. These are insights that go beyond just coding, but can you analyze a data set and describe what you're doing while you're coding and present the results to people in a meaningful way that are getting back to one of your earlier questions, that the cross collaboration is key.
What advice would you give to computational biologists who want their work to have clinical impact?
Well, definitely align yourselves with a company or a lab that has a clinical focus. I think more and more we're seeing the ability to translate genomics into actual clinical results. And it's not, to something we were talking about earlier, it's not just a matter of sequencing somebody's DNA and doing some variant calling anymore, right? I mean, there's a whole system and an organization of how do you interact with patients, how do you interact with healthcare systems, how do you interact with reimbursement, and being able to, something that I don't think I fully appreciated till I joined BillionToOne, was that this organization has that all. And this is why I think I'm feeling empowered to succeed because in a clinical context, whereas I thought I was doing clinically impactful work, and I probably was, right? But it was more futuristic research. But what I'm doing right now is impacting patients today. And that's very, very meaningful. So looking for an organization that is thinking holistically about all the different aspects of molecular diagnostics, not just the molecular part of molecular diagnostics.
What should trainees spend more time learning than they currently do?
I think understanding a lot of the science and engineering principles is going to be incredibly important, because coding as we know it today is starting to go by the wayside. It is somewhat of an identity crisis for bioinformaticians, but it means that understanding how software systems are architected. That's not going to go away, right? You still need to guide an AI to build the right tool in the right way. And making those decisions requires knowledge of how to build scalable software systems. There's a kind of an architectural engineering component that I think is going to become more and more important, as well as the science of things, right? Being able to continuously learn and adapt to the science of what you're doing will be essential in moving things forward. We're no longer just people who plumb and build pipelines. That's not what we do anymore.
If you could go back and give advice to your younger self, what would it be?
Don't underestimate serendipity.
And, to wrap things up, what are you most optimistic about in genomic medicine over the next decade?
Finding deleterious pathogenic variants and medicines that can overcome those deleterious mutations. That is an incredible change where first we had to develop the tech to identify mutations. Now we can identify mutations. And soon, not only can we identify these mutations, but we can proactively provide pharmacogenomic information and drugs that can mitigate those mutations. That's just an incredible place to be. We're seeing this in the prenatal space now, just starting to emerge with a couple of key examples. But that's a wonderful place to be, because now you can kind of complete the cycle of not just delivering challenging news, but ways in which you can mitigate those mutations.
Have any closing advice for earlier career scientists and bioinformaticians listening to the show?
Focus on what makes you happy, and curious. Stay curious. I think it's important to realize that what you learn today is not necessarily what you'll do in 5 or 10 years, but maintaining that ability to constantly learn and adapt is a fun place to be. And again, lean into the serendipity part. You never know when you're going to work on something and this particular project is going to be the extra meaningful thing. You didn't plan for it, but it turns out that the next 3 months are going to be an intense moment where you might learn an extraordinary amount in that short period of time that you didn't plan for, and you'll make a disproportionate impact by investing in those periods of time.
Elliott, thank you for joining us.
It's been my pleasure. Thanks for having me, Grant.
The Bioinformatics CRO provides computational biology services to academics, research institutions, biotechs, and pharmaceutical companies on a flexible hourly basis. Visit our website, bioinformaticscro.com, to learn more.